Good day. It's Thursday, September the 23rd. I'm Martin Gagel with Market Radius Research. Today we've got CEO Sherif Louis of Telo Genomics on the call with us. Telo Genomics is a biotech company pioneering the most comprehensive telomere platform in the industry with powerful diagnostic applications. I've got Telo Genomics on the call today for a few reasons. I've been following the company for several years, and it's really neat technology that seems to have some very beneficial health outcomes potentially. That's very fascinating. I also know some of the people behind the company with whom I have great respect and confidence. And full disclosure, I own a lot of stock of the company, so take it for what it is. I am biased. And remember, this is neither a recommendation nor investment advice. We're here to learn about the company. So Sherif, thank you for joining us today, and let's hear your story.
Thank you so much, Martin, and it's so exciting to be here today. We're always happy and ready to introduce our fascinating technology to new crowd and to update existing shareholders and investors and stakeholders about the progress that we're making to develop this technology into effective prognostic tools for disease and patient risk assessment. So without further ado, this is our forward-looking statement. This is just an intro slide for who I am. I've been with the company since 2015. I joined as Director Clinical Operations then. Before that, I was in the role of Vice President Scientific Programs with a national organization, Genome Prairie, which is a subsidiary of Genome Canada. I focused on fostering innovative, disruptive technologies, working with academia and industry to commercialize these technologies for a number of years. I have a technical background and I hold a doctoral degree, and my doctoral degree happened to be on the very early stage discovery of this particular technology that's being now commercialized by Telo Genomics. It's so thrilling to be doing this and watching and working on driving this technology, as they say, from the bench to the bed.
Sherif, just quickly, you mentioned Genome Prairie, which is part of Genome Canada. What kind of, is that a government kind of research agency, or is it a profit company? Just give us a very brief background on that.
It is a governmental organization funded predominantly by the federal government and also the provisional governments as well. It focuses on funding technologies focused on genomics in general across all life sciences sectors, like not particularly health only. They do a lot of agriculture and green energy, etc. They work through 6 arms spread across the country in Canada. There is the Ontario Genomic Institute in Ontario, Genome Prairie in the Prairies. There is Genome BC. It's one of the big centers actually for Genome Canada. It's very well funded. It's been over 20 years, organization surviving, and they are behind the commercialization of a ton of technologies across all life sciences sectors. And I was privileged to work with this organization for over 5 years. 3 of them, I was the Vice President Science Programs with Genome Prairie.
Okay, great. Thank you very much for that background.
No problem. So these are just a couple of pictures on where Telo Genomics is located. That's the Mars Center, downtown Toronto, University and College. And these are just a few shots for where the magic is being done. The wet lab, that's where all the wet lab workflow will done. This is our prep room here. That's the microscopy lab, has state-of-the-art microscopy technology, and also it houses the software platform, our proprietary software platform, Telo View. So all the microscopy and image processing and analysis happens in this room. And that is one of the stations, one of the 3 state-of-art microscopy stations that we do have, where we capture the images and do the image analysis. As you see, that's an image captured on the microscope, and it's being analyzed as this picture was taken.
Yeah, that's just how I was there about 2 weeks ago, actually,
Yes,
maybe 2 weeks ago,
absolutely.
right today. And just briefly, what is the Mars Center? It's sort of an innovation hub in Toronto, for those who don't know.
Exactly. It's called the Mars Discovery District, and it consists of three towers, south, east, and west towers. We are located at the south tower. The two out of 7, 8 floors are just innovation hub, or you call it an incubator for biotech technology startups and developing companies. We've been here since mid 2017. It's a state-of-art facility. We're privileged also to be here, and we're very functional location, I would say.
Thanks.
So I'm going to start off by telling you that the Telo View technology, or the technology that Telo Genomics is commercializing, has unique advantages, and it's not potentially will be a game changer, or will change the paradigm for prognostics for multiple myeloma, as I'm going to be describing, or any and several other cancers. This is already happening. This technology has a collection of advantages that put it ahead of the game compared to other technologies that are being developed or in the clinic. If one technology has advantage A and B, the other have C and D. Telo Genomics technology has all of the above, and I'm going to be walking you through some of these advantages. We stand tall on a very robust foundation of early preliminary and proof of concept, if you wish, clinical studies. Over 3,000 patients were already analyzed, 160 peer-reviewed publications all use this technology, and over $25 million in non-dilutive funding that were spent over 20 years in the academic lab as the technology developed from academia to industry to validate and de-risk the commercialization path, and every investment penny that comes to this technology. We stand tall compared to many other technologies with this foundation and strong and robust early on proof of concept studies, all of them clinical studies that were completed and published, I said. The company focuses always on addressing critical unmet need, critical clinical unmet need. We went into very rigorous due diligence process to identify our lead indication, which turned out to be the multiple myeloma disease, and the potential of this technology to provide prognostics tools, effective prognostics tools, and risk assessment tools to manage this disease. The technology is way ahead of the game using liquid biopsies, and I'm going to be stopping at that term in the coming few slides, which is the new paradigm and the new revolution in diagnostics and prognostics, because it is minimally invasive, very low invasive compared to traditional sample collection, biopsies, and what's not. As I said, we identified multiple myeloma as lead indication for us. We're looking here at a forecasted $4.6 billion spent on diagnostics and prognostics and pharma in the management of multiple myeloma by year 2027. It's a huge market, and the technology is getting ready for prime time. We partnership with the big guys. Our partnerships, as you will see, is with the Mayo Clinic, and that talks particularly to multiple myeloma. The Mayo Clinic in Rochester, in Minnesota, is one of the lead centers worldwide in the management of multiple myeloma. They have the largest collection of archived patient samples, and the clinicians that we are working with are the pioneers who are developing the guidelines for diagnostics and prognostics of this disease, as we'll see in the following slides. Before I dive into the technology, I just play a 1 minute or a 90 seconds maybe video clip that makes it easy for us to understand what is Telo View, what are we looking at, what are we measuring in an easy and interactive way, and then I will continue with the slides.
Telo View. Within our cells, safeguarding the chromosomes, are protective structures called telomeres, which maintain the genomic stability needed for healthy cell growth and vitality. Telomeric dysfunction leads to genomic instability, which can trigger disease, including cancer. Within a cell, the three-dimensional arrangement of telomeres, the cellular telomeric profile, is highly predictive of cell health and disease severity. The proprietary Telo View platform can detect the unique telomeric profiles for normal, pre-malignant, and diseased cells. To date, Telo View has been applied to 14 diseases. Telo View harnesses the predictive power of the telomere as a dynamic and universal biomarker. Telo View, genomic analysis for truly personalized medicine.
Basically, Telo View is a proprietary software that quantify key features in a very sensitive way. It's a multi-parametric assessment of features of telomeres. As the video showed, telomeres are the caps of chromosomes inside each cell, and as the literature and science tell us, it's a very well-established measure for genomic instability. And genomic instability is a very sensitive predictor for disease progression or stability. So in simple words, Telo View at the level of a single cell analysis, very sensitive compared to many other technologies, quantify these parameters, which informs on telomere health or dysfunction. And telomeres health or dysfunction informs on genomic instability, which predicts patients' risk, stability of a disease, progression of a disease, response to therapy. With this, the technology, as I said, the advantages are it's a very sensitive measurement. It goes down to the measurement of a single cell. It does not require more than 100 cells and does not require DNA amplification. And I'm saying this because several technologies in development, they depend on what we call NGS or next generation sequencing. The Telo View technology it does not include DNA isolation and amplification. It actually assess, as I said, on the level of a single cell, which in turn gets to be a lot of more faster and less cost and less number of patients required to achieve the same results that other technologies that focus on new generation sequencing will require. The technology inherently was built and validated using the liquid biopsy, whether it's blood or other fluids that can be with low or no invasiveness collected from the patient. The technology is validated for this on several cancers, including multiple myeloma as well, which provides faster, cheaper, painless way to monitor patients, can be repeated easily, and provides genomic analysis for each nature of the cancer. We get, I'm putting this slide because we get a lot of questions about, is technology applicable to liquid biopsy? And yes, it is. How can you compare it to new generation sequencing? So based on studies, preliminary studies, we say the technology is equivalent or even better in profiling cancer for the several advantages that I mentioned. If I will take a quick drive through our workflow, The workflow is very simple. Any sample that we can get our hands on, the technology is agnostic to sample type. As long as we can put it on a slide, we can process it, as we say. We do the telomere fish, which is fluorescence in situ hybridization. Has been a standard technique in the clinical laboratories for many years. It's trusted, it is sensitive. We do our three-dimensional imaging acquisition in the laboratory, as I showed you the picture earlier, and then we do the proprietary Telo View analysis, which gives a patient report saying this patient is high risk or low risk for this particular disease or indication that we are analyzing for. So that's in a nutshell, very simply and very quickly, the workflow that we run in the lab. That workflow is much quicker and simpler than many other technologies in the clinic or being developed as of now. We implemented, or on the way to implement, automation in each and every step of this workflow to minimize operator error, to increase scalability and high throughput as needed. Each and every step, as I said, is subject to automation, and some automation solutions and involving machine learning and artificial intelligence are already in place in identifying target cell for analysis or in processing the Telo View analysis itself. As I said, the workflow is automatable and can be easily translated into a high throughput as needed. The parameters that we measure, and I'll go through them very quickly. Many people, when they hear about telomeres and telomere analysis, they think that it's only about the length. And I'm happy to tell you that maybe several other technologies focus on telomeres length, but here we are doing a multi-parametric analysis. We quantify 6 different parameters about telomeres. One of them is length. Length is important, but it's not only the length. The number of telomeres in the cell, the fusion of telomeres, the shape of the nucleus where the telomeres and the chromosomes are housed, the position of these telomeres, and the overall volume. So it's a parametric analysis, and based on the quantification of these parameters, a decision or a suggestion or a report about a patient status, about the genomic instability of patient is developed. As mentioned on the first slide, the technology was validated in a number of indications, cancers, and neurodegenerative diseases. The company, as I said, focused now on multiple myeloma, but there are published clinical studies, different scales on all the cancers that are on the screen. As I said, this technology is a game changer in prognostics of cancer. We're starting with multiple myeloma, and in the coming several years, several other cancers will rely on this technology to for patient risk assessment and disease progression, as I said.
Sherif, sorry, can you just go back to the prior slide? Oh. And actually, the prior slide to this talked about the 6 parameters. So what you're basically saying with someone with multiple myeloma or one of those diseases, if they have certain parameters of length and telomeres and aggregates and all that, that kind of equates to, oh, you're at higher risk or maybe lower risk to this disease. Correct?
Exactly. That is the purpose of the clinical studies that we run. When we run clinical studies, we develop models that can predict patient outcome based on analyzing patient with known outcome at the beginning. This is the developmental process. So we analyze a number of patients that were good performers on a certain indication, and others who were bad performers, and we build a model that then will predict unknown patients at the single patient level. So that's the clinical studies that we run.
And is your technology the fact that other people, like you were saying, do the length, but the fact that you can measure all these AC ratios, nuclear positions, or is it you've developed some expertise, or is it more you've learned how to translate these signals into okay, this signal equals that, or probability towards that, and that signal is probability towards that. Where is your technology sort of within that framework?
It is two components, Martin, actually. It is two components. So the ability to quantify these parameters, that's proprietary. That's the Telo View software. That's the first component of Telo View software, just the quantification of these parameters in three-dimensional. Even several other technologies that will focus only on the telomeres length, they do that either by imaging two-dimensional images, or they do that by DNA amplification. And the results are so misleading because it does not represent the situation in a single cell. What we do here, we take three-dimensional imaging of cell by cell, and we measure all the 6 parameters. That's the first component. The second component is putting the measurements all together to make a meaningful report out of them. And that's the second component that is unique to each and every disease, and even to each and every test within a single disease. So the parameters will be measured, whether we are doing myeloma, smoldering, or drug response, as I'm going to be explaining later, or whether we're doing prostate. We're going to measure all the parameters, but the combination of measurements and thresholds and cutoffs that we develop during and through the clinical studies makes the second component. So it's both. It's the parameters and putting them together to make a meaningful report or result out of them.
With the detection or the sensing of these different parameters, is that like patent protected or just proprietary IP, or like can other people walk in and start doing this?
No. Actually, the technology is governed by a large portfolio of patents. All of them, maybe except one, are validated. We're talking here about 17 patents in Canada, in the US, and in Europe as well. We have two families of patents, let's say, core technology patents, and this govern the process of the measurements that we're talking about. And we have disease-specific patents. So we have core technologies, and those are like a family of patents, and we have disease-specific. So we have hematological disorders. That covers multiple myeloma, Hodgkin's lymphoma, leukemia. We have patents around prostate and analyzing circulating tumor cells. We have patents around Alzheimer's disease. So we have a large portfolio of patents governing this technology for approximately maybe almost 20 years still to go.
All right, thank you.
So I'll switch gears here to, as I said, to our lead indication, multiple myeloma. As I said, in 2019, went to a very rigorous process of due diligence to identify our lead indication, because there are proof of principle studies across the spectrum in all the cancers that I put forth. But myeloma was selected based on several parameters and several factors. The competitive landscape, the market, the clinical need, the existing scientific data that's already published. All that together made us feel that multiple myeloma is the correct lead indication for Telo Genomics to start putting out our first product that will be really a game changer and influence a decision-making change in the clinic. Multiple myeloma is a white blood cells cancer. It affects approximately 35 to 40,000 people every year in the US only. It occurs in plasma cells, which is a type of the white blood cells, as I said. Symptoms includes bone pain and bone fractures, of course, anemia, fatigue. Unfortunately, that disease is up till now is incurable. That is the maximum lifespan for patients diagnosed with multiple myeloma. It used to be 5 years. Now, with the development of new treatments, goes from 5 to 10 years approximately. As I said in the first slide, the North American diagnostic market for multiple myeloma is forecasted that based on market research reports, not our analysis. This is the third-party market research. And I have the references in the footnote at the end in the last slide. It's expected to grow to $4.7 billion in 2027. And we know that we're going to be part of this diagnostic regimen in the coming months and years.
And
so,
just to be clear, that 4.7 is just on the diagnostic side. It's not on the treatment of it. So,
no,
and you're only playing in the diagnostics. You're not actually any part of the treatment itself.
Oh, correct. $4.7 billion is a very small number for the treatment side.
Yeah.
Like this only diagnostics market. Diagnostics, prognostic. Usually when they talk about diagnostics, that cover diagnostics and prognostics. So the disease progression of multiple myeloma, it starts with a precursor called MGUS. These are very known stages of the disease for anyone who experienced that disease or have relatives with it. Very known terms. Starts with a precursor called MGUS that develops into another precursor, more serious precursor called smoldering multiple myeloma. And eventually the smoldering multiple myeloma transitioned to be an active multiple myeloma. The first two stages of the precursors, these patients are not treated. They are asymptomatic, no symptoms. It's just there are some indications in the blood analysis that tells that these patients are likely to develop multiple myeloma. Smoldering myeloma is much more high-risk precursor than MGUS, and almost 10 times progression comes from smoldering myeloma to active multiple myeloma. It's the smoldering transition to multiple myeloma is very difficult to predict. Patients without any indications, without any alarms, they transition to the active multiple myeloma. The clinic is looking for biomarkers, innovative, disruptive biomarkers to predict high-risk smoldering myeloma patients, and we can do this. We have published proof of concept studies that the technology has potential to predict these patients. And we are currently, as I'm going to be showing you, running clinical studies to validate and prove this. So the Telo View technology can contribute to the multiple myeloma management in identifying high-risk smoldering myeloma patients, in predicting the response of diagnosed myeloma patients to the first-line therapy, and even later on to monitor patients who already underwent treatment.
Sherif, sorry, just going back to that, just so I'm clear on how that is if you run the test on someone, and if it's a smoldering multiple myeloma, that is kind of a good thing. It's just sort of smoldering along. It's not actually causing damage. And then the fear is when it goes from smoldering to, I don't know, an active multiple myeloma. And so you detect whether it's in the smoldering state, which is kind of good, or whether it's likely or soon to go into a more active form of it.
Exactly. That's exactly the focus of our lead test for our lead indication. The way they call it in the clinic, and when we talk to those clinicians and medical doctors, they call them high-risk smoldering myeloma and low-risk smoldering myeloma. High-risk smoldering myeloma are the ones expected to progress to the full stage active myeloma soon, and soon to them meaning in less than 2 years. Low risk or stable smoldering myeloma are patients who are expected to stay in that stage for approximately 5 years or more. And that's actually the design of the clinical study that we are running with the Mayo Clinic, as I'm gonna show you.
Sorry, and just to ask again, so if you know it's a shorter term or likely a longer term multiple myeloma, the patient would probably be glad to hear that it's, let's say, the longer one or the shorter one, so they can kind of, there's less uncertainty in their lives. And then I would presume it also affects how the doctors treat it. If it's a long-term multiple myeloma, then maybe they just like, hey, leave well enough alone and we won't touch this. And if it's more of the shorter term one, then maybe we should get more active in the treatment.
Absolutely. That's exactly the key hot question in the management of multiple myeloma today. And we were just attending and participating in the International Myeloma Workshop in Vienna a couple of weeks ago. It's getting to be a couple of weeks ago, early September. And you see that the elite and the key leaders in the field, all their focus is on identifying these high-risk smoldering patients. Why? Because they want to treat them. Treatments are not easy, are harsh, are expensive, side effects. So they don't want to just treat all smoldering myeloma patients, because some of them can stay happily ever after till the end of their life at that stage. They want only to treat patients that are high risk, that will transition to the active stage of the disease within 2 years, but they do not know them. They cannot find them. With this technology, we have the potential, assessing telomeres dysfunction, assessing genomic instability, to pinpoint patients that will transition to active myeloma within 2 years. We have a published small size cohort that in a publication, very nice publication that came out earlier this year. We presented that also in the American Society of Hematology last year. And we are working with the Mayo Clinic, with a key clinician in the Mayo Clinic specialized in that particular niche, in the identifying of high-risk smoldering, to expand the study and develop a prognostic test. At the moment, they do some scores assessment based on blood work, standard blood work, and it's not taking them where they want to go. The message that we heard repeatedly in the International Myeloma Workshop last week or the week before, that they are looking and waiting for innovative genomics biomarkers to lead them through this process. And that's why a key elite clinician like Dr. Shaji Kumar out of the Mayo Clinic is working with us. The current scoring model that is being used, which is not giving them the message they want, is developed by him. And he's the key lead, he's the lead clinician on our clinical studies. Once we complete the studies and develop the test, the way that scoring model he developed is adopted, the way that test is going to be adopted. We are, as I said earlier, we are getting ready for prime time. We just need to complete these studies and develop the scoring model to identify on the patient level. We have already the proof of concept data. We just need to expand and finish the studies to develop this test.
So your test will have better predictivity, able to predict better at higher statistical rates than the current system. And then the next step with the treatment on the first line of defense of treatment, given the 3D telomere analysis, it's more personalized medicine where they say, oh, you've got this characteristics, this drug regime will probably work better for you, or this other drug regime. So you can sort of get to the better treatment option faster. Is that what you're saying?
It's a two-step process, let's say, Martin. The first step, you treat or not to treat. Like these patients, smoldering patients at this point, they're not being treated at all. So the first step is to say, who should we treat? And that's the first step, test that we are doing. Once you start treating, multiple myeloma has several regimens of treatment. And the nature of the disease, that patients receiving a specific treatment, they do well on it for a while, and then they stop doing well. They relapse, as we call them in a clinical term. They relapse. And the clinician need to change the regimen of the treatment. The technology, and that's another clinical study actually that we are running with Dr. Kumar, the second study, is to predict who of the patients receiving first-line treatment will respond and who will not. And again, within a time window, who's gonna be good on that treatment for 2 years for a year, and who's gonna relapse within 3 years. So it's a two-step, who to treat first, and number two, what to use for treatment, which regimen to use for treatment. And this is a clinical study that we're running with the Mayo, and the second one is also another clinical study that we are running with Mayo Clinic.
Thank you.
No problems. So as I said, like our priority test is identifying the high-risk smoldering patients. As I said, We keep hearing it in every meeting that we attend, with every discussion that we have with any of these clinicians. They have clinical studies with drugs ready to treat these high-risk patients if they know them, And it's the value offering for the technology to develop that test that will identify these patients early on. It's good news for the patient, of course. It should delay the progression to the full stage myeloma and would eliminate the risk of treating patients who doesn't need it. These drugs are very expensive. The side effects are very harsh. Identifying the right patient to receive the drug is key and important in the clinic. It is the most important question now based on my attendance to that workshop in Vienna. This is a key issue in myeloma that they're all gathering to find a solution for. And we are on the roll. We have the clinical study in progress with the Mayo Clinic, as I said. So just switching from science to a little bit of the economics, what market level are we thinking about when we say, okay, we developed the test, we are ready for the market now, and we're going to offer that test to the patients? What price level are we looking at? And at this stage, we are not much in a position to talk about, oh, we're going to have this price level or that one. But I'm putting in front of you several tests developed by several other diagnostics companies. All of them are actually in the market, in the clinic already. And this is the price range. I'm not making any comments where we're going to be. I'm just showing you this is the price range, the bracket of offering genomics testings. It's between $4,000 to up till $7,000.
And sorry, Sherif, just going back to that slide, how things are priced, it's not like, oh, you get a 50% or whatever kind of a margin. If you're able to, let's say, hey, you'll be better treated on this kind of chemo or that kind of chemo or whatever the treatment is, it could reduce the costs a lot for the insurance companies or the payers and have better outcomes. So it's kind of a value-based, it's based partially on value. If you can create $100,000 of value, you probably would have a chance to price it higher because it's such it avoids useless treatments or whatnot.
Absolutely, Martin. It's a formula really where, cost of treatment comes in place, cost of hospitalization comes in place. There are like few factors that comes in to assess the health economics value of each test. And you're absolutely right. The price range is not about cost plus profitability margin. It's about the value that the test can bring to the clinic, the amount of monetary savings, and also the management and the well-being of patients is also considered. This is a factor in that formula as well. How that test will be useful to predict patient outcome, improve patient management, and improve the overall lifestyle of patient, reach cure if possible, if you can identify patients that will get treated early or use the more precise treatment tailored for them. So it's several factors that comes into formula that will assess how much that test should be. You're absolutely right.
All right.
So when we think in Telo Genomics about the path to revenue, we have several actually paths that we can pursue, and we are keeping an eye and we are trying to be on the radar for all of these. Even we are in clinical studies stage. Of course, licensing or direct sale to pharmaceutical companies. Just going back to the points that we were discussing a couple of minutes ago, these type of tests are very important for drug developers. Identifying patients that will respond versus not. Sometimes you feel, pharmaceutical companies wouldn't want to know, but really when you see clinical study results, drug development clinical study results that did not come the way the pharmaceutical company was predicting, and the losses that they incur based on that, that they would want to be guided which group of patients they should be working with. And that can lead to what is so-called companion diagnostics, meaning a test attached to the treatment of or using that drug as a treatment modality, or could be in the clinical studies that the pharmaceutical companies are running. There could be also another path to revenue is licensing or selling the established diagnostic tests, like developed tests. That smoldering test can be licensed to either a diagnostic company or a laboratory company as on the level of a single test, or on the level of a disease, a group of tests. Like for multiple myeloma, we're talking about 3 tests here. There could be a direct sale, and we're working towards that eventually to develop the test and to have it in the clinic. We are working towards having a lab accreditation and designation to be able to offer the test to the clinic in Canada and in the US and even in Europe. And I will show in the last slide our initiative to cover this aspect or this path. There could be also the final one is direct sales in the shape of in vitro diagnostic test, and that's upon receiving an FDA approval. This is maybe the longest path to revenue, and if you can look at them 1, 2, 3, 4, that's how the path to revenue, the length that you can achieve this path is illustrated in that sequence. The fastest, easiest one is working with pharmaceutical companies. Following that is the licensing to existing laboratories or diagnostic companies as well, direct sales through a company reference lab, or obtaining the FDA approval. All these are options being considered as we speak for best and shortest and most efficient, highest profitability path to revenue. As I said, one of the key initiatives now that we are working on is achieving a laboratory accreditation. We are on the path to, we are already like in process of fulfilling the requirements to obtain an ISO 15189. We disclosed that in a press release a few weeks ago. We have consultants in place, we have a plan in place, and we are already in progress. It's not something that we are planning for. This is already in progress. We foresee that an accreditation ISO 15189, which is specific to medical laboratories, will be achieved in approximately 9 to 10 months. Latest would be a year. This is a huge evolution and transformational step for the company, where we present ourselves as an accredited lab with an ISO ready to work with all the large and big players in the field. R&D companies, you know, they have to pass through evaluation process, let's say, and obtaining a third-party accreditation like ISO is a certificate that we are on the quality level that appeals to industry, that appeals to the clinic. It makes a complete different presentation for the company trying to achieve partnerships, whether with pharma or with other laboratories. That's step 1. Step 2 is obtaining a license from the Clinical Laboratory Improvement Amendments, CLIA as it's known. So this is step 1, this is step 2. And we are in the process of step 1 already. As I said, we are looking forward to achieve this within 9 to 10 months, as I said.
Is CLIA a like an FDA type organization, or does FDA, like for the US, come into play on this? Like, who says, is there a third party that says, yes, this is statistically significant, or does they just let the market figure it out?
No, well, CLIA is an independent organization. It's not related to the FDA really, and it focuses on authorizing facilities like the lab facility and laboratory developed tests. So it authorizes a specific test for the use in that reference lab. The FDA approval is something that would allow you to have in vitro diagnostics test, and that's something that you can just spread in many labs if you wish. So these are very two different regulatory pathways. The CLIA is focused on the lab processes and the way the test is being developed and performed. FDA, they deal with the facts, with the scientific data only. Like CLIA, they will come and inspect your lab. FDA, they deal with the test or the drug dossier, and they made their assessment based on that. So it's very, very different.
All right.
Okay. These are actually the slides that I wanted to share with you. Finally, this is our leadership team. Dr. Sabina Mai, she's the director and she's a co-founder of the company. She is the chair of the Clinical and Scientific Advisory Board, and by all means, she is the primary discoverer of this technology, the primary innovator on all our patents, and she's an outstanding scientist, really overachiever, I would say, and we're all inspired with her science and dedication to science and to the improvement of patients. Chris is our CFO. He works out of Vancouver. And Guido, he's the chairman of the board. Very heavy acumen, business acumen, working with Roche for many years, working with other medical device companies for many years. A clear and a big veteran in the area of diagnostics, and we are very fortunate to have him on our board of directors as a chairman. This is the Clinical and Scientific Advisory Board members. As I said, it's chaired by Dr. Sabina Mai. We have Ken Anderson out of Dana-Farber, and Ken is the lead clinician in multiple myeloma. As I said, we were in that meeting that other week, and if you were there and you see how he is perceived and respected, I wouldn't say he is the Pope of multiple myeloma. I would say he's the God of multiple myeloma. And we are so fortunate to have him on our side. Dr. Anderson has been a clinical advisor for us since 2017. He sat with us on several calls, even board meetings, full days board meetings. We're so fortunate to have him on our side. We have Dr. Klotz. He is focused on prostate more. Dr. Hans Knischet is a long-term collaborator out of McGill. He is focused on multiple myeloma and Hodgkin's lymphoma as well. Dr. Drakenberg, he's focused on prostate cancer, same as Dr. Klotz. This is the share structure, and the data are, you can find it on CDOR or similar, just the breakdown of the PubCo information of the company for your reference. And for investor relations, please feel free to reach out to Tina or to myself at any time, and I'm happy to entertain questions. Thank you so much for your attention.
All right, thanks. You've got a lot going on here. You're doing the study with the Mayo Clinic. What should be, and you talked about getting the ISO designation in the next, I believe you said 9 to 12 months. Could you just give a bit of a timeline, let's say over the next 12 months or through 2022? What kind of news flow should we see coming out of the company?
Sure, Martin. So as like to start easy stuff, the ISO certification or accreditation, let's say, we look forward to achieve this within 9 to 10 months, and this is gonna be a gate opener for us for partnerships with other diagnostics companies or pharmaceutical companies on clinical studies, and that, as we said, could be a strong path to revenue. On the clinical development side, the studies with the Mayo Clinic are designed into phases and stages. So we have completed the very, very first stage, which is a feasibility stage. They send us a number of patient samples and we process them. We presented results to the lead clinician, Dr. Shaji Kumar, and we are in the process to receive or awaiting to receive a second batch of samples that will make a full stage. Full stage means it would lead to publications and announcements of results, etc. These studies, they are, as I said, like they get designed into stages. The full study design includes 3 stages. At the end of the 3rd stage, we have a clinical test. The full duration to finish those 3 stages, I would say it's between 15 to 18 months, maximum 2 years, I would say so. There are a lot of factors that we can control and other that we cannot control. For example, when COVID came, we could not control that. It slowed down clinical studies globally all over the world. We were fortunate to work with the folks in the Mayo Clinic to secure all the paperwork. Sometimes if problems happen like this, even COVID or even less than that, you could get delayed on your paperwork, the ethics approval, the institutional approval. We have all that secured. And we, as I said, we received a batch of patient samples that we processed, and we presented data already to the Mayo back. So we're waiting for the following stage, which is theoretically stage 1 after feasibility. This should, be completed within, I would say, 3 to 6 months at most. And there should be announcements at the time of receiving the samples, completion of the samples and results as well. Following stage, I would say another 6 months, and the final stage another 6 months. So let's say we'll spread them over 18 months, 6 months each.
So, and we've discussed this before, but what I think is kind of neat about, like, because the Mayo Clinic's sending you blood samples with people who have multiple myeloma, and you're trying to identify whether they're smoldering or whether they're going to get into a worse state. But they're not sending you fresh blood samples. They're sending you ones that are maybe 5 years old, and they already know who's become who's smoldered and who hasn't smoldered. Excuse my non-tech. You just do the test, send it back to them. They kind of match it up. Obviously, a little more complex than that, and they can give you the results right away. It's not like a therapeutic cancer drug where you got to wait 5 years to see what the 5-year results are on survivability. You get pretty fast turnaround on this.
Absolutely, Martin. Absolutely. We prefer always to run our clinical studies, as they say, retrospectively. Retrospectively means using archived patient samples where the associated clinical data and follow-up data are already in the database of the cancer center or the hospital that we work with. Like that, as you said, there is no wait time. We receive the samples, we process them. A statistician look at our results and at the clinical data. Like, it's a statistician that will do the math and identify the significant predictors. Later stages, as I said, will identify the scoring model, the formula that will predict the outcome of a single patient. So there is no wait time for clinical outcome. That's the beauty and the benefit and the advantage of running retrospective clinical studies. In 18 months, if we finish running these the samples of the 3 stages, we have a test. There is no wait for a follow-up data or patient outcome. This is all archived patients. Even when we say we're going to predict patients' outcome up till 5 years if they're stable, these patients are already older than 5 years. These samples and the data are already there.
All right. And I'm sorry, maybe you've explained, I just haven't got it. When you get this data, let's say if it's with the therapeutic, you'd submit it to the FDA and they'd look at the data. Do you have to do that same process, or do just the doctors and the clinics look at it and say, yeah, we like the results of this data? Or do you have to get sort of an FDA or the CLIA approval to say, yes, this is a valid multiple myeloma diagnostic and prognosticator?
Well, see, it's a few steps, but we don't need really to go to a regulator to, like, the short answer, we don't need to go to a regulator to put a test on the market. There is a path to present tests in the clinic under what they call it laboratory developed tests, LDTs. Based on the clinical data out of the clinical studies, which is vetted and reviewed by the clinicians, we publish the results of the clinical studies. And we publish the results of a validation study, which is exactly repeating the same thing on a different patient set to kind of confirm the results. Once you have these in place, you present the test to the clinic as a laboratory developed test. That is the purpose for us to run after getting the ISO 15189 and the CLIA designation. That at that point, with this accreditation, we are able to present and offer clinical tests that we developed in the laboratory, in the company's laboratory, to the clinic as laboratory developed tests, without needing to go to an FDA or getting a certain regulator to review that particular tests. Based on the clinical studies, the reviews, the publications, the validation, and the lab accreditation.
All right. And so you've got the multiple myeloma path going. You threw up a bunch of other cancers up there. I presume you'll be going after some other ones at a certain stage. Are you in that filtering process right now? Have you sort of shortlisted a couple where you think that they're the odds of you getting good diagnostic information or prognostic information there? And are you going to sort of talk about your next target in the next 6 or 12 or 9 months or anything like that?
For sure. We did due diligence on several of these indications that were on the slide, and we know which ones we should work on next, let's say, number 2 and number 3 and number 4. We just wanted to make sure we don't get distracted from having the first product out. We want to have the smoldering myeloma test out. We want to have the drug response test out. We want to have an MRD test out. Just getting a first product out will open for us the horizon to run several other indications. We want to maintain the focus, but we know which ones we should work on. A second, like a very close second, would be, for example, for sure, prostate cancer. Very strong data that we have, very strong publications that we have on prostate. So we have key opinion leaders around us. So for sure, a very close second would be prostate cancer. Another disease that is very, very important, and we have very strong scientific data, publications, market, would be Alzheimer's disease as well. Just as I said, like we went into a really very meticulous process to identify which is the lead indication we should focus on, and it happens to be multiple myeloma, and we feel that was a right decision, really, with all the factors and the conditions around us. We want to see that test, in the clinic, and the sky is going to be the limit.
So you're going to keep your eye almost 100% on the multiple myeloma. It sounds like maybe 18, 24 months until you starting to do some work on the next diagnostic. Is that a rough guesstimate?
Perhaps last time. As I said, like we want to make sure we don't get distracted from or slacking on having the first product out. But as the process develops and as we are getting closer there, and as opportunities present themselves to us in capital raises and whatnot, once we are comfortable that the multiple myeloma test is close there, we are happy to expand as well. Like a lot of economics comes to play too. We want to make sure that we don't open several clinical studies that we juggle them to complete. So, but for sure, like as far as due diligence, we know what we want to do next, and third and fourth.
All right. You have, and just we should wrap it up here. We're over an hour. Thank you, everyone, for your patience. You've got $4 million on your balance sheet. You've currently got $100,000 a month burn rate. That gives you a pretty good runway. As you get into these trials, is your burn rate going to increase, or should we just sort of expect we don't really have to worry about your runway at this point as your burn rate stays pretty low? Could you clarify that a bit?
For sure. Our burn rate will for sure increase a little bit. That doesn't make us worried about our runway. We're not looking at very massive spikes in our burn rate, but for sure, as the clinical studies develop and as we want to achieve things better and faster, we expect our burn rate to slightly increase for sure.
Okay. All right. Well, we should wrap it up here. Are there any final comments?
Well, thank you so much, Martin. It's a wonderful opportunity for us. We love to share our story and share our passion and excitement about it, share our belief and trust on the value that this technology is gonna bring to the clinic. We know this is it's not that it's gonna happen, it is already happening. We are working with the leads, and we are in progress, and we are so confident that we will develop tests that will influence decision making, a change in the decision making and in the management of multiple myeloma first, and several other cancers to come after that. Thank you so much for the opportunity, Martin.
Sherif, thank you very much. That was fascinating. Really appreciate it. Hopefully we'll get you back after we get some data out, and maybe we can discuss how things are progressing in a few months. So thank you very much, and have a great day, and hopefully we'll talk again soon.
Thank you so much, Martin.
Cheers.